Hello there, welcome to our video. We're going to cover DNA, RNA, and protein synthesis, and you've probably seen this already 10 times, so let's cue some jazzy music and make it less boring. This is by Adrianne, Arianna, and Michael, and it's voiced over by Arianna. And our first topic is DNA replication. Wow. So first we have to understand the representations we'll be using and the structure of DNA, which is a double helix model. And DNA's backbone is made of a phosphate group and deoxyribose sugar. There are 4 nitrogenous bases for DNA: adenine, thymine, guanine, and cytosine. And we've got a base pairing rule: adenine with thymine and guanine with cytosine. So the enzymes that are real catalysts in our processes today are helicase, the DNA or RNA polymerase, and the ligase, and we use different pictorial representations to represent what they'll do, and we'll see them working now. So starting DNA replication off, we have our enzyme of the helicase, which works as a zipper and separates the 2 strands and also creates a replication fork. So the DNA polymerase only adds nucleotides in one direction from the 5' end to the 3' end. So it creates 2 different directional strands. So for the leading strand, the DNA polymerase works towards the replication fork, which results in a continuous daughter strand. But for the lagging strand with the 3' end and the 5' end, the DNA polymerase has to work away from the replication fork, as it only adds nucleotides from 5' end to 3' end. So therefore, it results in fragmented daughter strand, which are called Ozaki fragments, and these are joined together by the DNA ligase. This is known as a semi-conservative process, as it keeps an original part of the strand but adds another new strand, so it ensures that accurate information is being passed down. So the next process is RNA transcription, or creating RNA. And first we're starting with initiation and promoters, which are regions of DNA with specific base sequences that can bind to RNA polymerase, start us off. And so the polymerase brings in the base pairs following the rules. And you'll notice that uracil is replaced with thymine in the new strand, as uracil is more durable and can withstand weather outside the nucleus. And so the DNA comes back together. So next we've got RNA processing, which is the editing of RNA. So the exons are what will leave the nucleus, and the introns are what stays in the nucleus, and this determines what's expressed. So we've got the 5' end cap and the 3' end poly A tail, which also determines how long it'll last. So we've got our helper proteins, which will cut and paste together the mRNA that is now edited and ready to leave the nucleus. But there are also 3 different types of RNA, not just mRNA. We've got tRNA and rRNA, which we'll talk about later. So the edited mRNA can now exit the nucleus and will now be used to do RNA translation, which is creating the protein strands. So starting with initiation, the start codon will bond to the ribosomes, and each codon is 3 base pairs, which is like a word that the ribosomes will translate. So the start codon bonds to the ribosomes at the A site, and here we'll give you a preview of elongation. So tRNA is charged with anticodon, which matches with the new codon. Also in the ribosomes is ribosomal RNA, which carries ribosomal proteins and carries out chemical reactions that join amino acids together. So now we're moving on to elongation, and this continues the process. The tRNA comes in and sort of stamps and bonds with the codons and creates this polypeptide chain, and yeah. Oh my god, mutation intermission. Mutations are a change or variation in the genetic material of a cell. These changes are heritable and can involve changes in the sequence of nucleotides in DNA or changes in the number or structure of chromosomes. Point mutations. So point mutations change a single base pair and occur at a single point. So here's a translation without many mutations. So it correctly transcribes things. The original strand is correct. So substitution is one example. It's when one of the base pairs is substituted for another. And sometimes it has an effect, but a lot of times it doesn't, as there are multiple ways to code for one amino acid. Next is insertion, where a base is added into the strand, and it basically frame shifts everything down, so it affects all of the coded amino acids after the inserted base. And in a similar vein, deletion has the same frame shifting idea, except a base is removed instead of inserted. There are also chromosomal mutations, which involve changes in the number or structure of chromosomes, and there are 4 different types: deletion, duplication, inversion, and translocation, which are all similar to what we talked about. But how are mutations affecting and how are they caused? So some are caused by natural events, just normal mistakes, or artificial means like DNA editing or GMOs. But some are caused by mutagens, which are chemical or physical agents in the environment. Chemical like pesticides or tobacco smoke, or physical like UV radiation. Mutations can be harmful and helpful. Helpful, for example, the mosquitoes who mutated to become resistant to the pesticides we used on them, and harmful, like the skin condition xeroderma pigmentosum, which is when your skin cells mutated and are unable to fix the cells damaged by UV light. And that's the end of weird transcription. Back to some normal transcription. We're in the termination stage, which occurs when the stop codon reaches the ribosome, which is UAA, and the protein releasing factor will release the tRNA and the polypeptide chain, and that'll result in the final product, which is protein. The end. Thank you for watching. Have a good day.